Okay. Okay. Hope you had a good weekend. So last time we had a tour of the cell where you saw all the organelles that plants have. Organelles are kind of the organs of a cell, as opposed to organs, which are kind of the organs of the whole plant. So today we're going to have a look at the plant organs, which are the leaves, the stem, the root, and if it has flowers, the flowers. Those are organs. Don't mix them up with organelles. People often do. Then another thing people often distinguish is the shoot from the root. So the shoot is the whole above ground part of the plant. So that would be the stem plus the leaves plus the flowers together is the shoot. Some other things you see in this picture, some terms that you may come across. For instance, a leaf often has kind of a stem of the leaf, the little thing that the leaf blade is sitting on that connects the leaf blade with the stem. That's called the petiole. And then the flat part of the leaf is called the blade. Also, often you see buds on plants which are young, non-developed leaves or flowers. So those are flower buds or leaf buds. The top of the stem, the tip of the stem is called the apex, which is in general a term for the tip or the top. So an apex of a mountain is the top of the mountain. And these buds are the place where new leaves or flowers appear. And there's a special name for the top of the plant, the top of the stem, where there is this beginning of a new leaf or flower, and that's called the apical meristem, where apical refers to the apex, and meristem is a dividing tissue. So that's where cells are dividing and new organs are being produced. Other buds that you find on other places in the plants, they're usually at the base of the petioles of leaves. They're called axillary buds or axillary meristems, which means secondary, not the primary, but secondary. Primary meristem or the apical meristem is the one on the top. And the apical one is usually the boss, you can say. If the apical one has a chance to grow, it may suppress the axillary meristems. And then you have strong apical dominance, as it's called. So if you have a sunflower with no branches, then the apical meristem suppresses the side branches from growing. But other plants are more branched, so it depends on the species to what extent these secondary meristems are suppressed. And hormones control this, as you will see in section 3. So the place where these leaves come out of the stem is also called the node. Node means like a knot. It's often a thickening of the stem. So the node is the place where things come out of the stem. Leaves and also buds start there. And the place in between two nodes is called the internode. So you have node, internode, node, internode. So every node you have leaves coming out. And it's relevant to know these things because if you know where these growing points are, particularly these meristems, where dividing tissues are, if you want to clonally propagate plants, meaning you want to just make a cutting and grow it into a new plant, you have to know where the plant is able to grow a new plant from, because only from those meristems, so the axillary and primary meristems, can plants grow. So if you take that cutting and you pamper it, you can grow it into a new plant. But you cannot take an internode and just stick it in water. It will not grow a new plant because there is no dividing tissue in there, unless you treat it with special hormones, so kind of artificial. Then you can take any plant cell and grow it into a new plant. But it's just a natural plant without messing with its hormone levels. You have to have a meristem, a dividing tissue, in order for it to grow it asexually into a new plant. So I think those are about all the organ parts that we see here. Vegetative shoots, vegetative and reproductive is also another term that people use. The vegetative stage of the plant is when there are no flowers yet. And when it starts to grow flowers, then it's in the reproductive stage. So we also talk about reproductive tissues or structures versus vegetative structures. The leaves will be vegetative structures, flowers are reproductive structure. So reproduction is the plant reproduces itself into a new plant. Oh, and if we look at the roots, then we see sometimes that there's a thicker main root, which is called the taproot, and then there are lateral roots, which are side roots. You see that particularly in dicot plants like sunflower beans, whereas grasses tend to have more fibrous roots, where all the roots are kind of equal in thickness. So they're a little bit different that way. Also, the leaf shape, leaf orientation is different between monocots, grasses, and dicots, where monocots tend to have their leaves a little bit more upright, and they don't have really so much an upside and a downside, whereas dicot plants often have more horizontal leaves with really an upper side and a lower side, which are anatomically different. So if we look at the leaf as the first organ that we zoom in on, then we see it has the three typical tissue types that all plant organs have, which is the dermal tissue, the ground tissue, and the vascular tissue. The dermal tissue, that means skin. Dermis is skin. That is the outside layer of the organ, and it's often covered with a waxy layer of molecules which protects the organ from drying out and also from attack by pathogens. And that's called the cuticle. So that's the waxy layer. This is the dermal tissue, and it's called also the epidermis. It is different from the rest of the leaf because of the cuticle, but also in its metabolic activity, it doesn't have any chloroplasts, for instance, except for only these stomata. Here at the bottom, you see this hole, which is enclosed by two cells, and that is called a stoma, which means mouth in Greek. And you already saw the plural of Greek can be, you add ta at the end, like plasmodesma, plasmodesmata. Here you see another example, stoma, stomata. So that's plural. So it means mouth, and it really looks like a mouth under a microscope. And all the epidermal tissues of the shoot have these. They are for gas exchange. And the plant can open and close them at will. If it's really dry, it will keep its stomata closed. And if it's moist and there's light, then it opens them. And that tells you what the point is. It opens them to let CO2 in for photosynthesis. So if there's no light, then the plant cannot do photosynthesis, so might as well keep its stomata closed. Because terrestrial plants always have to struggle with needing to take up gas, but losing water in the process. If it opens its stomata, water molecules are going to evaporate through the stomata. So there's a cost of having the stomata open. On the one hand, the losing of water has also a function in sucking up water from the roots, so it's a good thing too. But still, plants always has to be careful not losing too much water. That's a bit of a struggle. So stomata are the places for gas exchange. And they are actually the only cells of the epidermis that have chloroplasts. They have a little bit different metabolism. So that's the epidermis. And in dicot plants, the stomata are at the bottom of the leaf, which makes sense. That's the shaded side. So there, if you open your stomata on the shaded side, you lose relatively less water than if you open them on the sunny side. So that makes sense. But grasses that have more vertical leaves, they don't really have such a difference in stomatal distribution. And aquatic plants actually have their stomata on the top if they have floating leaves, which makes sense because that's where the air is. So usually it makes sense. So that's the epidermis. Yeah, question?
Did you say that there's no chloroplasts in the epidermis, but there are in the stomata?
That's what I said. So stomata are in the epidermis are only in the guard cells that surround the stoma. So the guard cell is the term for the two cells that kind of guard the stoma, two kind of sentries. And as you will see, opening and closing of stomata is an osmotic effect. And what that means is they can be blown up with water, or they can lose that water. And that opens them or closes them. So that's the dermal tissue. Then the ground tissue or filler tissue is also called parenchyma, and that's regardless of which organ we're talking about. And in the leaf, it's called mesophyll. So those are the parenchyma is kind of the cell type, which is kind of a nondescript average cell. Nothing special about it. Parenchyma, filler tissue. But in the leaf we call it mesophyll. And phyll refers to leaf. Meso means in the middle. So it's the filler tissue in the middle of the leaf. And the mesophyll serves as a function to photosynthesize. And you see that because it's full of green dots, and those are the chloroplasts. And they capture the light and they make sugars. And that feeds the rest of the plant. These sugars are exported from the leaf by means of the vascular tissue, which you see here as these red and blue, particularly the blue vascular bundles. The blue bundles are called the phloem, which is a type of vascular tissue that transfers water with sugars and whatever else plants wants to export from photosynthesizing leaves to the rest of the plants that needs to be fed. And then the red vascular tissue that you see here, that's called xylem with an X, and that serves to bring water and inorganic nutrients mainly from the roots to the shoots. So there are two types of vascular tissue, xylem and phloem, and you find them actually in all organs. And they have the two separate functions. You will see a lot more about these vascular tissues also in one of the next slides. A little bit more about the mesophyll. Especially in dicots, you see a clear difference in the tissue that is on the top of the leaf versus the mesophyll tissue at the bottom of the leaf. You see at the top of the leaf they're very tightly packed, the cells, no big holes in between them, and their shape is kind of narrow and tall. That's called the palisade parenchyma. And they capture the bulk of the sunlight. They're very efficient at doing that because they're so tightly packed. And then the bottom part of the mesophyll, that is called the spongy mesophyll. And the reason why they have such big air holes in between them is to facilitate gas exchange, so to bring CO2 to all the cells, so including the palisade parenchyma. Because most of the gas comes from the bottom, it's good to have a lot of space, airspace, in the bottom of the leaf. So spongy, it's clear, it looks kind of like a sponge, big air holes. And it also makes sense to have relatively fewer cells per surface area in the bottom of the leaf because there's simply less light left. After the palisade parenchyma is done with it, the light intensity has become much lower because the light comes from the top. So you don't need as many cells there to capture the rest of the sunlight. That's about all for the leaf. And this slide shows you a little bit the structure of these vascular cells, if you can call them that, vascular fibers or bundles. We have on the left the xylem and on the right the phloem. So the xylem was the red vascular tissue that you saw here. So that brought water and inorganic nutrients from the roots to the shoot. And then on the right of the next slide is a close-up of the phloem cells, which are the blue vascular tissue which brings water with sugars and metabolites from leaves to other plant parts. So left xylem, that's red, that was red, and on the right phloem, which was blue. A difference between the two is that xylem is actually dead at the time when it becomes functional. So it's just cell wall left standing. It's like a straw, really. It's like a sturdy structure, a hollow fiber that's just cell wall left standing, and it's very fortified cell wall. You often see spirals like this of secondary lignin material, secondary cell wall material, or rings, and it all serves to fortify. And that is because xylem is under a lot of actually negative pressure, tension. The xylem transports fluid by means of suction, and that suction is created by transpiration from the shoot, and that sucks up the water from the roots. And if you have a very flimsy straw and you try to suck really hard on it, it may collapse. Maybe you've had that, bad quality straw. So you don't want, the plant doesn't want that happening, so it has a really fortified xylem to withstand that tension. The phloem, on the other hand, which brings water, sugars, etc. from leaves to other plant parts, is actually under positive pressure. It loads sugars into the phloem at the leaf end, water will follow, because water tends to always follow solutes. That's osmosis, and you'll see more about that in the next module. So this creates a positive pressure, and that pushes the water through the phloem. So if you would puncture a hole in phloem, it would come out. But if you puncture a hole in xylem, air would be sucked in, or water, whatever is on the outside. That's the difference between them. And the other difference I mentioned, xylem is dead cells that used to be living, but then the plant sacrifices them so that it's more efficient for transport if you just have the cell wall left standing. But the phloem is still alive. However, the phloem is very adapted to be very good at transporting molecules. It has a lot of plasmodesmata at its ends, and that's what you see here. It kind of looks like a sieve, and that's why it's called a sieve plate, or they're called sieve cells. They have so many plasmodesmata it looks like a sieve. And another adaptation that you see particularly in angiosperms, which is here on the right, is that actually the phloem cells have lost some of their organelles and some of their functions. They could not survive by themselves. That's why they have helper cells, which are called companion cells. So little helper cells next to the phloem cells kind of feed them and do some of their functions for them. In gymnosperms, so like pine trees, they are not that advanced. They're a little bit simpler. But they're also, all these cells, these phloem cells, are alive. And on the left side in the xylem, you also saw gymnosperm structures on the left and angiosperms on the right. And again, the angiosperm ones are a little bit more derived, a little more complex than the gymnosperm xylem vessels. So phloem vessels, xylem vessels, those are some terms that people use. Sieve elements or vessel elements. And in the case of these phloem cells with helper cells, this is also called the sieve element companion cell complex. Sometimes people call that SECC. Just so you know what people are talking about if you read this somewhere. So xylem and phloem we find everywhere in the plants. All organs have it. And phloem is also fortified, not as much as the xylem cells, but they're also pretty thick. So that was leaves, yes? Question?
Are the companion cells then are completely specific to angiosperms and not found in gymnosperms?
Yes, as far as I know, yeah. The companion cell, the question was, are companion cells specific for angiosperms and not gymnosperms?
And they help with structure, you said?
The function of the companion cell is to feed the sieve element. So the cell that is the actual phloem that transports water, sugars, etc. It needs to be fed because it has given up some of its functions, and that's what the function is of the companion cell. Okay. Yeah. So that was the leaf, and now we have a look at the stem, and next we'll look at the root. And the stem, again, has those same basic tissues. And by the way, what a tissue is, it's a group of cells that work together. They have the same function. They look anatomically the same. They are in the same organ. Then we call it a tissue. So it's a bunch of cells that work together in an organ. So in the stem, we have, again, the dermal tissue or epidermis, and that's actually this outer layer. This arrow, this line should stop here. Should stop here at the edge. It's just the outer layer, epidermis. Then we have the filler tissue, parenchyma cells. So that's the dermal. That's the filler tissue, parenchyma. And then we have the vascular tissue. And the vascular tissue depends on whether it's a dicot or a monocot. If it's a dicot, then the vascular tissue is arranged in a ring. And that's how you can recognize the dicot. Whereas in monocots, the vascular tissue is kind of interspersed everywhere throughout the stem. And no matter if it's a monocot or a dicot, the vascular components, xylem and phloem, are always arranged in such a way that the xylem is toward the interior of the stem and the phloem is toward the exterior. And in the middle, there is, in dicots, a layer of dividing cells, which is called the vascular cambium or a meristem. That's dividing cells, and they create more of this phloem and xylem. So this is a way by which means the stems of dicots can grow in girth, so in thickness. And dicots have that more than monocots. Monocots don't tend to have this secondary growth in width, so that grasses tend to stay pretty narrow. You don't have a grass that looks like a tree. So that has to do with the arrangement of this of the vascular tissue and this cambium that is present in dicots. And then there are names for the filler tissue that is outside of this ring of vascular tissue. It's called the cortex, versus the filler tissue that's within this ring, and it's called the pith, so the pith and the cortex. So dermal tissue, ground tissue, or filler tissue, and vascular tissue. So that's the stem. And then the root. So if we look at this cross-section of a root, we see again the epidermis, so dermal tissue, then we have the ground tissue, or filler tissue, made up of parenchyma cells. And then we have the vascular tissue in the center. So you can see whether an organ is a root or a stem, because if it's a root, then the vascular tissue is just a central cylinder, versus in the stem, it's usually a ring, or it's a bunch of interspersed vascular structures if it's a monocot. And if we zoom in on the central cylinder where the vascular tissue is in the roots, then we see that there is kind of an X-like structure, or a plus. That's where the xylem is, and you see here it's red, so that's consequent with what you saw earlier in terms of coloration. If it's red, it's xylem. If it's blue, it's phloem. So the X is xylem, and then the rest of the circle is the phloem. So vascular tissue, xylem, and phloem are always side by side, and there is a function to that, because they can exchange water and also other molecules, but particularly water can flow from xylem to phloem and back, and this plays a role in water movement in the plant. Around this central cylinder where the vascular tissue is, we have a ring of cells, or two rings of cells. And first the pericycle, and then the endodermis. So endodermis versus epidermis. Epidermis is the dermal tissue on the edge that surrounds the entire organ, but the endodermis is kind of another layer of skin, but it's within the organ. And the function of this endodermis, as you will see when we look at transport and of water and nutrients, is to separate the vascular cylinder from the outside world, because the xylem here, particularly the xylem, is dead tissue. So technically it's apoplast. So if there were no barrier around the vascular tissue of the root, then this apoplast of the xylem would be in direct connection with the soil, basically. Because everything can freely flow through cell walls. So through the cell walls, anything, nutrients, water, whatever, can flow freely all the way up to the central cylinder. If there were no barrier there, then there would be no control over what goes to the shoots and what stays there. So plants would leak nutrients that it wants to concentrate, and it might actually take up toxic elements that it doesn't want. So having a layer around vascular tissue helps the plant separate itself, its body, from the outside world. So we'll see this endodermis later. It's a special cell layer which has an impermeable cell wall. So it has a little kind of a corky layer. Maybe you've heard of the Casparian strip. Kind of has a raincoat on, so things cannot go through there. It's impermeable to water flow. Endodermis and its cell wall. So what we see in other things here is that there are these protrusions from the epidermis. Those are root hairs. They serve to increase the root surface area, so it can scavenge better for nutrients and water. It can take it up more efficiently. If we look at this longitudinal section here on the right, you see the central cylinder, again, as purple, and another term for that is a stele. And then around it, this filler tissue is also called the cortex for roots. And then you have the epidermis. At the tip of the roots, there's the apical meristem. So there's an apical meristem at the tip of the shoot, but also at the tip of the root, where cells divide. and that's where more cells are formed. And after these cells are formed, they can start to grow, and that happens further away from the tip. That's called the elongation zone here, where cells grow. And plant cells typically grow by elongating, meaning they get longer in one direction rather than growing in all directions. And that has to do with those cellulose fibers being all arranged in parallel, remember, so they can just grow in one direction. So zone of elongation, that's where you could actually measure the growth if you had a little ruler. And then there's the zone of maturation, where the cells mature, so they reach their final shape and function. That's the root. I already mentioned these meristems, the points of growth. And let's have another look. So we have the apical meristem of the shoot at the top. We have axillary meristems as buds at the base of the petioles, at the nodes. And then we have vascular meristem or vascular cambium, which is in between the phloem and the xylem, and which gives growth in girth and thickness. And the root we also have apical meristem, and then we can have axillary meristems to give the side roots. So if you want to asexually reproduce plants, or if the plant wants to asexually reproduce itself, then you have to have at least one meristem there that can grow then into a new plant. So you can make a cutting, put it in water, and it will grow a new plant. So if you would look at this shoot, for instance, you could cut right here. You cut at an internode, cut, 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 and then you make sure you have always this one meristem, and that will then grow a new branch. And then it will spontaneously form new roots. So asexual reproduction, what happens in all these meristems is that cells divide. All the DNA is first duplicated, and then it's separated over two daughter cells, and then the cell divides through the middle. And then you have two daughter cells, which are genetically identical to each other and to the parent cell. So asexual reproduction creates more of the same, genetically exactly the same as the parent. Or you could also say every plant part is also genetically identical. All cells have the same DNA. And that's where flowers come in, because being genetically the same from generation to generation is not a good idea, because the environment changes, is unpredictable. So it's good to have some genetic variation in your offspring, then you have more chance that one of them is going to survive if you have unpredictable environmental conditions. So sexual reproduction creates genetic diversity, genetic variation. And what happens in sexual reproduction is that the genes are shuffled, kind of like a deck of cards. The way the genes are arranged is that if you have, for instance, 24 chromosomes in a certain plant species, then there are 12 sets of 2. So there are 2 copies of every chromosome. So if you have 24 total, that means you have 12 different ones and 2 of each. And another term for that is diploid. So mature plants and also animals, they are diploid, so 2 copies of every chromosome, and these can be reshuffled. So what happens is that special sex cells are made, pollen and egg cells, and they get only 1 copy of every chromosome. And this is a random process, so it's total chance which copy they get. So you can get many different gametes, different egg cells and pollen cells, and they are genetically different depending on their kind of luck of the draw, which copy did they get. So you have many, many different gametes coming from one organism, and they can then be rearranged in all these different combinations. Even if you have sexual reproduction from one plant that produces both pollen and egg cells, like a lot of plants do, if it sexually reproduces with itself, then it can create an enormous diversity of offspring. And what you often see with plants is that as long as things go great, then they often do asexual reproduction, because what's the point of creating diversity if things are going great? But if the plant is stressed, it often starts to flower, and then it's going to make seeds, because then it chooses to do sexual reproduction, because if things are changing, then it's a good time to create some diversity. And also seeds are a good way to survive stressful conditions. They're very protected from harsh conditions. They can survive for years, droughts, whatever. So that's another reason why plants tend to do that. So what happens in sexual reproduction is that the diploid organism has a special kind of cell division called meiosis, where the genes are separated. Out of every 2 copies that the plant has, only one ends up in each of the daughter cells, which are called the gametes, the pollen and the egg cells, and they are haploid. So they have only 1 copy of every chromosome instead of 2. And then these pollen and egg cells find each other, they fuse together, and then you have a diploid cell again. And this first cell is called the zygote, which has just formed from a pollen and egg cell fertilizing each other, and then you have 1 diploid cell, which then proceeds to perform the regular mitosis, the kind of normal kind of cell division, where all of the daughter cells are genetically identical. So from that original diploid cells, all the thousands of cells that are in the eventual mature plants are all genetically the same. They come from mitosis from this original zygote. So meiosis is only used for sexual reproduction. Offspring has half the number of chromosomes, half the number of genes. Then you get fertilization. You go back to being diploid. And after that, via mitosis, 1 cell grows into an entire mature organism. So that happens, the making of the gametes, and the fertilization happens in the reproductive structures of the plant, which you see here as a flower. And what you have is the leaf-like structures, the sepals down closest to the stem, and then you have the petals, which are often pretty because they are meant to attract pollinators, if they're insect pollinated or animal pollinated in general. And then you have the actual reproductive parts of the flower. There's the male and the female parts, which is called the carpel and the stamen. And the carpel consists of the ovary, the style, and the stigma, where the stigma is the kind of the tip, and that's where the pollen cell is going to land. And the pollen is produced on the anthers, which are on a filament and together form the stamen. And the pollen can be collected by insects or other animals from here, or the wind can also disperse it. Pollen lands on the stigma, and then it starts to grow down here, and then pollen and egg cell find each other, and then you get fertilization. Depending on the plant species, sometimes plants can have self-fertilization, sometimes they have mechanisms to avoid it. So in that case, if they cannot fertilize themselves, that creates even bigger diversity. If they can only fertilize another organism, another individual. So that was kind of a recap of material that you may have had in earlier classes about plant anatomy, cell anatomy, some basic functions. Any questions about this first module? Okay, so that's the first module. Close that one. The second module is also, I hope, to some degree, a bit of a recap from lower division courses. And what we're looking at is more processes in plants, some basic principles of processes of reproduction, of gene expression, and of metabolism. So that's the topic here. Kind of setting the stage that later when we start talking about specific processes, you already know the vocabulary and some basic principles. So we're going to have a look at genomes, at proteins, how they're produced, how this is regulated, and about what these proteins do, so enzymes, metabolism. So the genome, that is the DNA, the genetic material that is present in cells. And in plants, the DNA is in 3 organelles. By far, the most of it is in the nucleus, which is shown here on the right. And it's arranged as it is typically for eukaryotes in a number of linear molecules. And the number is always the same for a certain species. So you can always say, well, 2n equals 24 for a plant species. Then it has 24 linear DNA molecules, which would be 2 times 12, because 2 copies of every chromosome. In addition to this bulk of the DNA, there are two other organelles that have a little bit of DNA of themselves. You already saw this before in Module 1. Those are the chloroplast and the mitochondrion. They have a circular DNA molecule, which is actually a remnant of their life as a prokaryote. This is typical for bacteria. They have that kind of genome organization as a circle. And they make some of their own proteins. So that's where we find the DNA. And regardless where the DNA is, the central dogma holds true, which says DNA is transcribed into messenger RNA, which is then translated into proteins. So that's called the central dogma. The process where DNA is copied into a messenger RNA is called transcription, which means you make a copy, you write a copy. That's exactly what happens. A copy is made of a piece of DNA, and we go from one kind of nucleic acid, which is DNA, to another kind of nucleic acid, which is RNA. They're very similar, but they have small differences. And whereas the DNA is a gigantic molecule, the mRNA is a nice, compact little molecule that can move around. And that RNA, messenger RNA, functions as a message for how to build a protein. It goes to the location where proteins are made, which are the ribosomes. And depending on where the DNA is, the ribosome may be in the organelle, the chloroplast or the mitochondrion, but most of the messenger RNA in the cell are translated on ribosomes that are hanging out in the cytosol, and they can then produce their protein either as free ribosomes in the cytosol or by binding to the ER, depending on where the final destination of the protein is, whether it's part of the endomembrane system or not. So that's where the message, the messenger RNA, 3 nucleotides are translated into one particular amino acid. There are 20 different amino acids in proteins. So depending on the arrangement of the nucleotide, the order of nucleotide, A, U, G, C, can be any combination, you get a different protein. And the genetic code, like which 3 nucleotides encode for which amino acid, is universal in life on Earth, which reflects our common origin. We all come from the same source. We all speak the same genetic language. I also sometimes compare this to the DNA is in the library. Yeah, it's like you have all these recipes for how to cook things, but you cannot check out the cookbook. So if you want to cook something, the furnace is the ribosome. You have to make a copy first in the library, and the copy is the messenger RNA. You can then take that copy to the stove, which is the ribosome, and then you can make the protein. And the recipe is the messenger RNA. So I mentioned in Module 1 that every cell has the same genetic material, the same DNA, but only a subset of the genes is actually active, is expressed, as it's called. Only maybe 5 or 10,000 out of 25 or 35,000 genes are on. And this gene expression is regulated, and we'll see in this chapter some modes of regulation. So transcription is shown with, or the DNA, where the DNA is transcribed is shown with red stars, and then where the translation happens is shown with green stars. So then you get the proteins, so they're made on the ribosomes, and the proteins are the actual workers in the cell that make things happen. So they are the busybodies. They're making, for instance, they're doing metabolism. They're producing metabolites, turning one metabolite into another metabolite. We call them enzymes. If proteins have a catalytic function to speed up a biochemical reaction in a cell, then they're enzymes. And other proteins serve, for instance, as transporters. So they sit in membranes, and they specifically transport things across that membrane. And specific is an important word here. Transporters are specific. They don't transport just anything, but only one or maybe 2 things. And enzymes, similarly, don't just catalyze any reaction, but only 1 or maybe 2 specific reactions. And that is because these proteins, these transporters and these enzymes have binding sites where they bind the molecule that they either transport or that they convert into something else. And the term for the thing that they bind is called substrate. So they have a substrate binding site, which is very specific. It's like kind of a key in a lock. So the enzyme has, or the membrane transporter has a binding site, kind of like a lock, which only binds specifically the substrate that it acts on, that it transports or that it converts into something else. And because enzymes and transporters are so specific, the cell needs to have tons and tons of them. So there's a very large fraction of the genome that has these functions, transporter and enzymes. What you see here, this kind of looks like maybe like traffic in a city. And it's kind of like that. What we're looking at is metabolism. And the black dots are the metabolites. So these are molecules that can be turned into one another. And the lines that connect these dots are the metabolic processes that turn one molecule into another molecule. And typically, each of these processes is mediated by an enzyme. So that gives you an appreciation for how many different enzymes there must be. There must be thousands of different enzymes. And all of these enzymes can be regulated in terms of how many molecules of those enzymes there are, but also how active each enzyme is. They can often be turned on and off. So this gives an enormous degree of flexibility, of response to the environment. So there's a lot of homeostasis in cell metabolism where the cell can all the time fine-tune itself, respond to changing conditions, to keep all the metabolite levels optimal so that things don't get out of control. So it's a delicate balance. What you see here in the center, this kind of looks like a yo-yo. That is the respiratory pathway, respiration. Remember that happens in mitochondria where sugars are broken down to CO2 and water. That has a very central position in cell metabolism. So we'll pay some special attention to respiration. So metabolism is the sum of all the biochemical reactions that happen in an organism or in a cell. And those reactions can either be building up something or breaking down something, and there are terms for that. If you build larger molecules out of smaller ones, we call it anabolic reactions. So there are biosynthesis reactions. And catabolic reactions break down larger molecules into smaller ones. So respiration, where sugars are broken down into CO2 and water, is an example of a catabolic reaction, whereas photosynthesis, where you build sugars out of CO2 and water, is an example of an anabolic reaction, where you build something. And as you can imagine, catabolic reactions release energy. If you break down large things into small things, that releases energy. But if you have to build something out of bricks, say, build a building, then you have to put energy into it. So that's the same for anabolism and catabolism. Catabolism tends to produce energy. Anabolism requires energy. And that is a good thing for the cell because what it can do is it can actually harvest, capture energy that comes from catabolic reactions. That's where ATP comes in, which can temporarily store that energy. And then it's a mobile molecule. It can go to another place where an anabolic reaction needs to happen. And then the ATP can release that energy again and make that anabolic reaction happen. So ATP is a very important metabolite molecule in cells. It's a mobile form of energy. And another important function which couples different reactions together is the function of transporting electrons. And electrons often go hand in hand with protons. And the kind of reactions where these electrons are shuttled about are called redox reactions. I already mentioned those before. So if you have two molecules and one wants to get rid of some electrons and the other one wants those electrons, they can react, electrons move, and then we have a redox reaction. One molecule loses electrons, it's being oxidized. The other one gets electrons, it's being reduced. Electrons are negatively charged particles. They're very tiny particles, they have no mass, but you can consider them tiny particles. They have a negative charge. So where electrons are transferred, then the one molecule gets more negative, the other one gets more positive. And often protons go hand in hand with the electrons. What a proton is, it's basically a hydrogen atom that has lost its electron, because whereas all atoms except for hydrogen have protons and neutrons in its nucleus around which electrons are orbiting all the time, hydrogen is the only atom that has only a proton and then an electron going around it. So if it loses its electron, you're left with just a proton. So that's also a very, very small particle. And protons and electrons often are shuttled about in the cell from one molecule to the other. This is important in metabolism. And the cell has come up with some transport mechanisms, molecules that can use protons or kind of load protons and electrons as a cargo and move them about. They're mobile, and then they can pick them up where they are given off by a molecule, and then they can bring them to another place where they are then used again. So an example, for instance, if you have photosynthesis, then we have water, we have CO2, they are combined to form sugar. But that's not the way it happens in the cell. What you have is you have water, which gives up protons and electrons. They're given to a molecule, which is NADP, which can accept the protons and electrons, turn into NADPH, then it can move to where the CO2 is, and then it gives those protons and electrons to the CO2, and then you get the sugar. So even though the equation says CO2 and water react, that's not the case. There's something in the middle that carries the protons and electrons back and forth. So NADP or NAD is a molecule that can do that, and FAD is another one. So they function in, for instance, photosynthesis, but also in respiration, because there you have the same thing. Sugar doesn't react with oxygen. Then you would get flames, but it goes in small steps, and we have intermediate molecules that shuttle the energy about and that shuttle the protons and electrons about. This shows you what ATP looks like, and one thing that is interesting to point out is that ATP has two important functions. One is it is a building block to build RNA, so messenger RNA has it, ribosomal RNA has it. It's one of the 4 nucleotides that make up RNA, but its other very important function is it's a little package of energy. ATP stands for adenosine triphosphate, and tri means 3. It has 3 phosphate groups, which are packed very closely together. You see that here, 1, 2, 3. Phosphate group is PO43-. They're negatively charged. And as you know, equal charged groups, molecules, repel one another, right? So you have to put quite a bit of energy into this molecule to pack those 3 negatively charged groups so closely together. This little part of the molecule is harboring a lot of energy. It's like a spring. So if you release this bond, then it really jumps away. You can imagine this is an energy-rich molecule. So this ATP can give up a phosphate group, and then it turns into ADP and P. So adenosine diphosphate, it only has 2 phosphates, and then there's a single phosphate, which is P or PI. People write it often, but it's a phosphate group. It can also lose 2 at a time, but usually it's 1. So then you have ATP. It gets hydrolyzed, so it reacts with the water molecule, and then you get ADP and P, which is a phosphate group, and some energy. And the amount of energy is shown here, 7.3 kilocalories per mole. If you remember what a mole is, Avogadro's number, it's 6 times 10 to the 23 molecules. So a lot of molecules. And kilocalorie is a unit of energy. Another unit of energy is the kilojoule or the joule, where 4.2 joule is 1 calorie. And this particular reaction of ATP splitting up into ADP and P releases 7.3 kilocalories per mole. If you go the other way, ADP and P are combined to form an ATP. That requires 7.3 kilocalories per mole. So this is an important concept. Every reaction is reversible, and the energetics involved with going in one direction is equal to the amount of energy involved in going in the other direction. But in one case, the energy comes out of the reaction. In the other case, you have to put it into the reaction. This is a principle that holds true for all chemistry. So this is the amount of energy that you can store in a mole of ATP. As you produce it, then you can carry it around the cell and use it wherever you want. Like membrane transport, moving things along microtubules, or having 2 reagents react with one another and forming a new molecule. Okay, guys, that's it for today. Have a nice Monday.